In this section, we'll be taking a close look at microscopy. When we think about the macroscopic world, the dimensions that we consider are given in centimeters and meters. You're probably fairly familiar with those. However, in microbiology, the world now becomes much smaller. The largest we'll look at are millimeters. Then we consider micrometers and nanometers, so 1,000 times smaller than the millimeter. The smallest bacteria that we'll consider are 200 nanometers. And the largest that we'll consider in the microbial world are the large protozoans. They're about 4 millimeters in length. Of course, those are visible to the naked eye because it's about half a centimeter, or a quarter inch if you need that conversion. Viruses, however, are very, very small. Small enough to infect bacteria as well as these protozoans. So they range from 20 nanometers to 800 nanometers. Now, some viruses then are slightly larger than the smallest of bacteria, but most of them are not much bigger than the largest of our macromolecules. So most of the microorganisms that we'll be dealing with, we can see with a light microscope. However, there are a few that we'll not be able to see unless we were to have an electron microscope. We'll look at electron microscopes later on in the chapter. The bottom line here is that we wouldn't be able to view this world without an essential tool, which is called the microscope. First, we'll look at the light microscope and understand a little more detail. Remember, Leeuwenhoek was the first to design a microscope. He used a single lens variety, and then Hooke devised the compound microscope. We'll take a little closer look at what this means here shortly. So there are two key characteristics of microscopes. First of all, magnification, and second, the resolving power. Magnification results when visible light waves pass through a curved lens. So for example, when you put a hand lens magnifying glass or a spherical glass ball in front of an object, it will appear larger. This is because light experiences refraction as it passes through a curved lens. The image is formed by the refracted light when the object is placed a certain distance from the lens and is illuminated with light. So light is a key factor here. Now, what is refraction? Refraction is simply the bending or changing of the angle of the light. As we change the angle of the light, we're projecting an image on our eyeball that is larger. This image is enlarged to a particular degree, and we quantify that degree by the power of magnification, like it would be a 4 times magnification or a 10 times magnification lens. Refraction is the basic principle behind all optical or light microscopes. Now here's a microscope that might look much like a microscope that you'll be using for your lab work this semester. However, the first ones were much more simple than this. They had one lens, like a magnifying glass, and Leeuwenhoek's first scope was based off this one lens concept. Today we have a compound light microscope. That's what you see in this slide. The additions that were made to a compound light microscope were a second lens system, a lamp that can illuminate the specimen, and then a condenser, which focuses the rays of light onto the specimen. Now, you might be asking how all of this works, so let's take a closer look. Magnification is going to occur in 2 phases. We have 2 lenses, one called the objective, which is nearest the specimen, right here, and then a second lens that's closer to the eye, called the ocular lens. Magnification then occurs in 2 phases. The first lens, the objective lens, the one closest to the specimen, produces a real image. And then that image is projected onto the second lens, the ocular lens, the one that's close to the eye, and it forms a virtual image. So you're really seeing an image of an image. And this image can only be seen well if there's adequate magnification, resolution, and good contrast. We'll discuss the concept of resolution here shortly. There are generally 4 different objective lenses, sometimes 3, on the nose piece of the microscope. Okay, we corrected that spelling. And they range from 4 times to 100 times. 100 times is the oil immersion lens. That requires oil. We'll cover that shortly. And then the ocular lens is most often a 10 times lens, sometimes 20 times, but most often 10. So in order to determine the power of magnification, we're going to multiply the objective lens times the ocular lens. So any given scope could begin with 4 times 10, which would give us 40 times magnification. So your object would appear 40 times larger than it would to your naked eye. Or 100 times 10, so 1,000 times. If you happen to have an ocular lens that was 20 times, then of course this number could be 2,000 times. So potentially in a light microscope, you could see your specimen at 2,000 times what you could see it at with your naked eye. So now let's take a closer look at what resolution means. Resolution is the ability to distinguish two adjacent objects or points from one another. In humans, our resolution is about 0.2 millimeters. The ability of a lens to resolve is called its resolving power. Now, here's an equation that you don't really need to know, but just to give you an idea of how we calculate resolving power. It has to do with a function of the wavelength of the light, measured in nanometers, divided by 2 times the numerical aperture of the objective lens. Now, you might be saying, what on earth is numerical aperture? We don't really need to worry about that. It's fixed for each lens, and it's between 0.1 and 1.25. And there's a whole bunch of mathematics behind it that we're not going to worry about. So we'll just keep that as a fixed value. And then the wavelength of the light, we know that the light source for the microscope contains many wavelengths of visible light. So the numerical aperture describes the relative efficiency of a lens in bending the light rays. So this fixed value of 0.1 to 1.25. And shorter wavelengths are going to provide a better resolution or higher resolving power. So as I said, humans can resolve to 0.2 millimeters, and the light microscope can resolve to 0.2 microns. The oil immersion lens is a higher magnification lens, so it has the highest numerical aperture, about 1.25. And with a resolving power of 0.2 microns, some of the smallest bacteria and viruses simply can't be seen with this scope, so we'd move on to the electron microscope. Let's see if we can understand resolution a little more closely. As we increase the magnification, let's say we go to the oil immersion lens or the 40 times lens, we're going to decrease the resolution of the image. So we can adjust the amount of light entering the condenser using an adjustable iris or diaphragm. So less light, or we can even create special dyes that help increase the resolution at higher magnifications. In addition to that, shorter wavelengths of light improve the resolution. If you look at the image below here, long wavelengths of light are represented by the large yellow circles, and smaller wavelengths of light are represented by the smaller yellow particles. So shorter wavelengths allow light waves to pass through the smaller spaces because they occupy less space. Thus, we can produce a more detailed image. As you can see with the hand here, the smaller particles or the smaller wavelengths of light can move more clearly between the fingers, and thus we get a full image of the hand. Less resolving power comes from having longer wavelength light that can't quite occupy these smaller, tighter spaces, and thus we get a less resolved image. So an oil immersion lens allows us to view things at a much higher magnification, but without the oil we would have refractive loss, which would result in a much less resolved image. Because oil has the same optical qualities as glass, it prevents refractive loss. It's as if the specimen is embedded in glass all the way through the lens. Because oil has the same qualities as glass, it prevents refractive loss. You'll notice that the side without oil, some of the light that passes through the specimen is scattered into the air, and some of it is scattered onto the glass slide. However, with the oil side, all of the light goes up in through the lens. The scattering of light that we see in the non-oil side decreases the resolution. So oil increases the resolution and also increases the numerical aperture. Let's take a quick concept check. Why do we use immersion oil with our 100th time objective lenses? Is it to reduce the light scatter? Is it to reduce illumination, reduce magnification, or reduce contrast? Well, we just covered that one. So A, reduce light scatter is the correct answer. Visible light microscopes or optical microscopes are microscopes that use visible light. So in the visible light spectrum, from the short wavelengths of blue up until the longer red wavelength, there are 4 types of optical microscopes: the bright field, the dark field, phase contrast, and interference. We'll look at all of them in a little more detail here. In addition to optical microscopes, there are other microscopes that include fluorescence microscopes, confocal microscopes, as well as electron microscopes. Let's begin by looking at the bright field microscope. This is the predominant way we'll be viewing things in this class. It's the most widely used. It forms its image when light is transmitted through the specimen. The specimen produces an image that's much darker than the surrounding illuminated field. And this can be used with live, unstained, and preserved or stained specimens. In dark field, we're using a regular bright field microscope, but a disc is added, or a stop, that blocks the light from entering the objective lens. Now all we're left with is peripheral light that's reflected off of the specimen. So it's effective for visualizing living cells that would be distorted by drying or heat, or that cannot be stained with the usual methods. However, it doesn't allow for the visualization of the fine internal details of the cell. Next, phase contrast microscopy. This transforms really subtle changes in light waves that pass through the specimen into differences in light intensity. So imagine that you had a beaker full of similar clear objects. Because they have similar optical qualities, you can't really distinguish them from each other. This is very similar to how organelles are inside cells. So what this type of scope does is it transforms very slight differences in absorption that we cannot perceive into differences in light intensity that we can, thus allowing visualization. This technique is very useful for viewing the intracellular structures such as bacterial spores or granules or the organelles inside a cell. Now, differential interference microscopy is like phase contrast, but has some additional refinements. For example, it has a couple of prisms that add contrasting colors, and it allows for a detailed view that's somewhat three-dimensional, very colorful image. And we can get this different view even in unstained specimens. Now, fluorescence microscopy includes a UV radiation source and a filter to protect the viewer's eyes from these dangerous kinds of rays. We use this technique with dyes that show fluorescence, hence the name of the technique. There are ultraviolet rays that allow visualization of this fluorescence and form colored images against a black field, depending on which stains are used. We can use this to diagnose infections that are caused by specific bacteria, protozoans, and viruses, but we have to use fluorescent antibodies for that. In confocal microscopy, we can view cells at higher magnifications, and we use a laser beam of light to scan the various depths of the specimen. So this alleviates the issue that we'll run into with a light microscope about focusing at different planes, because they're three-dimensional structures, but we're only getting a two-dimensional view. In confocal microscopy, we're allowed a three-dimensional view because the beam of light scans at various depths in the specimen and produces more of a three-dimensional image for us. It's most often used on fluorescently stained specimens. Now, finally, to get the most detailed images possible, we have electron microscopy. Scanning electron microscopy allows creation of a three-dimensional image with very, very detailed views. You can see some of those in the images over here on the right. The specimens are not scanned by electrons, but they're coated in a metal and then bombarded with electrons. The scope will scan back and forth and then pick up the energy of the electrons as they're deflected off of the surface of the specimen. That electron pattern is detected and then it's picked up and displayed on a monitor screen. The wavelength here that we're able to use is 6 picometers, which is 100,000 times less than the red visible light that we can see. This is why we're offered so much better resolution with scanning electron microscopy. So, a quick concept check. What wavelengths of light would give the best resolution when viewed directly with your eyes? Red, yellow, violet, or ultraviolet? Red is 650 nanometers, and ultraviolet is 260 nanometers. And we did learn that it's the shortest wavelengths of light that give the best resolution. So that would leave us down in the ultraviolet region, right? However, the key here is with your eyes. Ultraviolet light is not something that we can see, so violet would be the correct answer in this case.